So this is the introduction to the cardiovascular system lab. The reason that we're covering a portion of the cardiovascular system at this moment is that we are about to learn about the liver, the first organ you will study in the course. And in order to be able to understand the blood flow in the liver, you need to have a basic understanding of the different types of blood vessels that are found in the body. The remainder of the cardiovascular system, the heart, will be studied in the spring in coordination with your dissection of the heart in gross anatomy and your study of the cardiovascular system in physiology. So our goal here is to help you identify different types of blood vessels that you encounter in any tissue or organ that we look at in this course. The first type of blood vessel is the largest artery that is found in the body, and that is an elastic artery. So this is a bit of an unusual stain, and it's a relatively low magnification image of an aorta. And because of the way that it is stained, you don't really see any of the cellular detail. But what we want you to understand is that every blood vessel is made up of at least one or two layers, and often three layers, that you will need to be able to identify. So starting from the lumen, which is here, the first layer you encounter is the tunica intima. And then below that you have the tunica media, which is the widest extent of this blood vessel. And then out here you have connective tissue, and this connective tissue is called the tunica adventitia. What's unusual about this, or one of the unusual things about this light micrograph, is that the extensive elastic membranes do not take up any of the dye. And so everything that stains blue is cellular material, but it's hard to pick out the nuclei of the smooth muscle cells and the cytoplasm of the smooth muscle cells in between here. This is very low magnification, so even there is not that much detail to be seen. But what you should understand is that an elastic artery is filled throughout its tunica media with layers upon layer of elastic membranes, and these provide elastic recoil against the high blood pressures that are exerted when the ventricles contract and push blood out of the heart. So now we're looking at a very low magnification light micrograph where we're looking at anatomical structures. This is to help you orient towards how blood vessels are arranged in the body. So this sample was taken from a cadaver, and up at the top and down below, these polygonal structures are part of skeletal muscle, which you'll be studying in block number 3. In between, supplying the muscle and also supplying other tissue in the area, are two quite large blood vessels. The smaller of the two is called a muscular artery, which we'll look at in the next slide. And next to it, as is very common, runs a vein. Now, since this was taken from a cadaver and blood pools in veins after death, that is why you see so much blood here and almost no blood here in the artery. Outside of these two, you can see a number of branches, and in each case what we're looking at are arteries which are usually smaller in diameter, running with veins which are most often larger in diameter. The largest artery, which we looked at in the preceding slide, is now shown at medium magnification in this light micrograph. So the lumen of the blood vessel is filled with plasma, which has clotted, and the layers of the walls of the artery can be seen now more clearly. These flattened nuclei at the surface are the endothelial cells, and those cells plus the thin connective tissue layer underneath are called the tunica intima. And then you'll see a pink line which is running and is very coiled in appearance. This is an internal elastic membrane, and this is made by the smooth muscle cells that are in the tunica media. And then you have a number of layers of smooth muscle nuclei going out to another pink line, which is the external elastic membrane, also made by the smooth muscle. And then beyond that external elastic membrane is the connective tissue layer called the tunica adventitia, and how this connective tissue blends from the layer of the blood vessel into the surrounding connective tissue is never easily seen, and you should not be concerned about that. Just understand that there are three layers: tunica intima, tunica media, and tunica adventitia, and the presence of the internal and external elastic membranes indicate that this is a muscular artery. And also we use the characterization of the number of layers of smooth muscle to distinguish this muscular artery from an arteriole. By comparison, the adjacent vein, which is shown at higher magnification here, is the lumen is full of clotted blood, and the nucleated cells that you see here are the lymphocytes and granulocytes that we will study shortly as that are part of the immune system. Note that large veins also have one or two layers of smooth muscle, which are characterized by which you can identify by their elongated cigar-shaped appearance. There is also a tunica intima and a tunica adventitia. For the veins, the tunica adventitia is the largest or thickest layer of the three. The largest veins can have some elastin in their tunica media as well. So here's a medium magnification light micrograph. At the upper left, we have a lumen, which does not contain any obvious blood cells, but it has a tunica intima and a tunica media that is approximately 1, 2, 3, 4, maybe 5 cells in thickness, then goes into a tunica adventitia. So this is a muscular artery or arteriole. And then over here we have just a thin layer, which is the tunica intima with an occasional smooth muscle cell, and this would be the vein or venule that is associated with this artery or arteriole. And here we can also see scattered in the connective tissue some of the smaller blood vessels. So here is an artery which is wrapped by two layers of smooth muscle, which are defined by these elongated nuclei. So this is an arteriole, and it has an internal elastic membrane, which is this pale staining region, and then it has an endothelium, which forms the tunica intima. The other vessels that you can see here are an even smaller blood vessel, and here is a smooth muscle cell that wraps the entire thing. And note that this smooth muscle cell is actually contracted in a way that occludes the lumen. So this is the smallest of the arterioles with one layer. It is sometimes called a met arteriole, and its function is actually to control blood flow into the capillary beds. So by comparison, what is a capillary? Well, here's a very small blood vessel right here with just an endothelial cell. So this would be an example of a capillary. And you can see that there are a variety of sizes of blood vessels based on their lumen, and also based upon whether they have smooth muscle in the tunica media or not. And then over here, in between, we have a different type of vessel. This is indeed not a blood vessel. This is a lymphatic vessel. And it is most easily characterized by the spacing of its endothelial cell nuclei. So these two are endothelial cell nuclei, these two are endothelial cell nuclei, but you see that there exist large gaps before you see other endothelial cell nuclei, and this is to permit interstitial fluid, which has come out of the blood and into the connective tissue space, to be resorbed into the lymphatic vessels, which will then ultimately bring the fluid and deposit it back into the blood. Note this structure right here, which is an infolding of the endothelium or tunica intima, and these two flaps form what is called a valve, and the valve is necessary to prevent backflow of fluid within the vessel. Valves are found in veins and occasionally in large venules, but mostly in veins, and always in lymphatics, because there is no smooth muscle of any kind around lymphatics that will aid in the progression of that fluid back towards the heart. And finally, you should understand that with blood vessels, that is, arteries and veins, or arterioles and venules, run two other structures. One is a lymphatic vessel, and the other is nervous tissue. So this is a section at medium magnification, a light micrograph. And at the top here, we see two blood vessels that are fairly large in diameter. And then we also over here see some other blood vessels which are much smaller in diameter. And then we see a vessel which seems to be filled with pale staining fluid. And then to the left of that, we see a structure that you learned to identify as nerve in the first block. So here we have two larger blood vessels. By comparison, here is probably a venule, and next to the venule is a vessel containing fluid, which is the lymph. So you have arteries or arterioles and venules, and then lymphatics, and also nerves. These happen to be fairly large bundles of nerves running with these blood and lymphatic vessels. The smallest vessels we see here are capillaries, and it is the fluid from these capillaries that then is collected in the lymphatic vessels to be returned to the blood. So what I'd like you to do now is to continue and understand the remainder of the cardiovascular system lab that focuses on blood and lymphatic vessels. And then I'd like you to go to Learn at UW and take a self-assessment quiz to identify different vessel types. And then finally, with in your small group, get together with your friends and look at some of the unlabeled images to test your understanding of the blood vessels of the cardiovascular system.